And all of these individuals have pathogenic or likely pathogenic variants in this transcription factor, FOXI3. All of them inherited from the parents, and most of the parents were not affected. So again, a seemingly dominant phenotype with a reduced penetrance. To put this into perspective, from the 670 individuals that we studied, 21 had mutations, bad mutations in the FOXI3, and that is 3.1%. So we open a window to the multiplicity of genes and their respective proteins that are involved in the development of that particular organ that we call internal and external ear. So it is an important opening to the developmental pathway of how to make an ear. Here's the panorama of the variants in a schematic of this particular protein. The protein has about 450 amino acids. It has one domain that's called the nuclear localization domain that tells the protein to go to the nucleus and act as a transcription factor, and has one big domain here that is called fork head domain, with which this transcription factor binds to the DNA that regulates. I will not bother you with functional analysis. All of the variants have reduced activity in a luciferase assay, and the variants that were in the nuclear localization domain, the majority of them, they produce a protein that mislocalizes to the cytoplasm and not to the nucleus, shown here for the aficionados. I will not bother you with a 3D reconstruction. Now, how to solve the problem of penetrance? We came across a case report in the literature. So most people poo-poo the K reports. However, sometimes they are very important in understanding a phenotype. So this colleague, Giorgio Gimelli, who worked in Genova, has described this case with his abnormal ear, and has described a small deletion on chromosome 2 that contained the FOXI3 gene. And at the time, he made the hypothesis that this FOXI3 gene, among all the other ones that were deleted, they're important for this particular phenotype. So I asked Gimelli to restudy this family. And the hypothesis is that this affected individual has the mutation, the deletion here, and the father also has the deletion, but the father had no abnormality. So that, to me and to others, it ringed a bell that perhaps the modifier or the important aspect of the penetrance is the nature of the other allele of FOXI3. Because as you know very well, when you inherit the variant, the mutation from the father, you do not inherit the other allele too. You inherit the other allele from the mother. So this individual inherits the deleted allele from the father, but this allele inherits from the mother, which is different from the other paternal allele. So we sequence the remaining allele, this one, in that individual, 2.5 megabases worth of sequence, because we made the hypothesis that the remaining allele contains variability that, if in trans with the mutant allele, gives you the phenotype. And in essence, the so-called dominant inheritance is also recessive, and it has to do with both alleles of FOXI3. So when we sequence that particular allele, what did we find? We find a totally normal sequence that is present in the population. It's a sequence of about 70 kilobases that contains 2 linkage disequilibrium blocks, 2 pieces of DNA that are inherited as blocks from one generation to another and from many generations to others. And it is marked with polymorphic sites that you and I have in our DNAs. This allele is quite common in you and I. In South Asians, the Chinese, is this particular allele here. It's about 19%, yes. And in Europeans, that's the third one, and the frequency is 12%. So here's a common allele that perhaps in trans with a mutation gives you the phenotype, which is remarkable because this phenotype, the abnormal ear, is perhaps due to the combination of a rare and a common allele at the same locus. And usually when people do exome diagnostics or genome diagnostics, we look at rare variants and we don't pay that much attention to the common variants that co-segregate with the rare one. The conundrum is also solved, semi-solved, with this particular family that's of Greek ancestry. So here's an individual that has an abnormal external ear, this lady. You can see the ear before the reconstruction and after reconstruction. And she has a variant, a mutation, of an arginine 240 to cysteine. But the mutation in FOXI3, the mutation is also present in her two brothers. This one has a mutation, and this one has the mutation. And the mutation is also present in the unaffected father here. So all three males have that particular variant, but they don't have external abnormal ears, as you see here. They don't have this because, that's also a hypothesis that is similar to the previous one, because they inherit, they have a different transallele from the affected individual. The affected individual has inherited in trans the blue allele from the mother, while the two carriers but normal, they inherited the green allele from the mother. And of course the father has yet a different allele from the blue one. So if you do this analysis in all the affecteds, you see that the majority of the affecteds have the blue allele as a trans with the FOXI3 variant, either the total area of block 1 and block 2 of ligand disequilibrium, or the second part or the first part. And if you do the statistics of all the 21 affected pedigrees, the p-value is quite respectable, 10 to the minus 5. Of course, one needs additional cases to nail down this. But for the moment, all the evidence is that a common variant in trans with a rare variant in FOXI3 will give you that phenotype. Can we do a mouse with it? We created a mouse. Andy Groves at the Baylor Institute in Houston, he has created a knockout mouse for the FOXI3, homozygous. And the knockout mouse does not have at all external ears. So it goes with the possibility that FOXI3 is an important gene for the development of the ear. And then a mutant mouse was made with the human homologue of the arginine 240 to histidine, and this mouse did not have any phenotype in heterozygosity and homozygosity. Any phenotype in the ear. It had additional phenotypes in the immune system. And then we said with our Chinese colleagues, Yong Biao Zhang and Kei Mao, to make a triple mutant in the mouse, a mutant that has 3 different variants, one after the other, in the same allele. The variants were found in human cases too. So that particular mouse, in homozygosity, and some phenotypes in heterozygosity too, has abnormalities in the facial bones that I will not show you in details yet, and also had no further normal development of the external ear. And the other amazing thing is that these mice, they also had craniofacial or hemifacial microsomia. You see the mouth here of this mouse, and the mouth of this embryo mouse, that's a normal, that's a mutant. So half of the face is not well developed, and several of the bones are not very well developed, as expected for this particular triple mutant. So the hypothesis, the mechanism. The mechanism of this developmental abnormality is that the combination of rare and common variants in the same gene in trans cause the phenotype, and that is the mechanism of the so-called reduced penetrance in this phenotype. So if you have a variant, a mutation in both alleles, you have the microtia. If you have one variant in one allele and the common haplotype, the permissive haplotype in the other allele, you have the phenotype. If you have a deletion in one allele and the permissive variants in the other, you have a phenotype. In contrast, if you have a bad variant in one but not the permissive allele in the other, you have a mild or no phenotype. Or if you have a deletion in one allele and a non-permissive haplotype, you also have a mild or no phenotype. And that example is one of the very few examples that open the way to make the emphasis of the penetrance or the nature of the trans allele. And you've heard another phenotype this morning of the child with hematuria. And this one smells to me that one needs to look at the trans haplotype of the variants in the collagen 4 gene. And the project that we do now is we try to understand the regulation of that particular haplotype. What are the EQTLs, or the sites in the haplotype that are associated with high expression or low expression of the FOXI3 gene? And you see the hypothesis here. You have a mutation in one allele and a very high expressor haplotype on the other. You produce mostly the normal protein, FOXI3. If, however, you have the mutation in one allele and a low expression haplotype on the other, you have mostly the expression of the mutant allele and not the normal. And we have identified EQTLs, that means single site polymorphic sites, binding sites for different transcription factors that during the mouse development in day 4 or 5, they have a differential expression of FOXI3, and that could explain the differential phenotypic expression of that trait. But for that, in the future. I thank you very much for your attention.
Thank you, Professor Antonarakis.
For those of you who were here yesterday, I promised to show you this book that the Swiss Institute of Bioinformatics made for me for the sequence of chromosome 21. So I'll give it to you. Mr. Chairman and Mr. Chairman, I can have questions. This is chromosome 21, 4 kilograms, and you see the sequence. It's letter 7 of this particular thing. I give it to you to look at it, but at the end, please return this to me.
Professor Antonarakis, thank you for a very excellent presentation showing how one transcription factor can control the development of an ear. Is there time for one quick question from the audience? Yes.
So thank you for another great presentation today. I'd like to ask, how would you explain the asymmetry in the phenotype? Right. How come we only have the problem on one side?
Yes. Of course, I wish I had the answer. That's a mystery. Not every case is asymmetric. There are cases with symmetric, and as you see in the photographs, some individuals have a problem in both ears, and others in one or the two. There are several hypotheses that you can make. The most plausible hypothesis is that as every transcription factor, the expression level is very low. So transcription factors usually, they make 4 to 6 RNA transcripts per cell. And not only this, but for genes of low expression, like the transcription factors, only one of the two alleles fires. So in other words, the cell only expresses one of the two, each cell. And the idea is that the few cells that are light up and they are destined to make an external ear in that one day window in the mouse development and a few 3 days windows in our development, then stochastically, in one side it could express the normal allele, and in the other side could express the mutant allele, or in both sides express the mutant allele, and you have the phenotype. This is very difficult to study for FOXI3 because of the small window of expression, but we're trying to look into this in mice too.
I think we need to continue. So once again, thank you, Professor Antonorakis.
Thank you.
I would like to call again the chairs of the next presentation, Dr. Manolis Nikolous and Dr. Stefanos Christodoulidis.
Thank you very much. So we would like to call Professor Paul Moss for the next lecture. Professor Moss, who I know very well, I was really honored to work with him in Birmingham, is the Professor of Hematology and Deputy Head of the College of Medicine at the University of Birmingham, UK. He runs a specialist clinical service in chronic lymphocytic leukemia and was co-author of Essential Hematology, one of the best-selling hematology textbooks, which we also use at AUC for our hematology course. He runs an immunology research group with a focus on viral cancer and transplant immunology, and is chief investigator of the UK Coronavirus Immunology Consortium, a consortium of 20 universities which leads the UK immunology response to the COVID pandemic. He holds program grant funding from the Medical Research Council and Cancer Research UK, and several of his discoveries have been translated into clinical trials, including a novel form of antigen-specific cell therapy. He has published over 230 publications with over 28,000 citations. Professor Moss was awarded an OBE in the Queen's Honours List of 2022. I would say rather delayed from his achievements. And we would really like to welcome Professor Moss with a lecture title From Diagnostics to Therapeutics, Antibodies Take Center Stage in COVID-19 Pandemic. So thank you, Paul.
Thank you, Manos. That was a very generous introduction. Thank you very much indeed. And to Ioannis and everyone for invitation to this incredible conference. I must say, quality of the science today has been incredible. So yes, as Manos says, I'd like to talk to you today about the topics of antibodies in COVID-19. We haven't had too much on COVID, thankfully, have we, in the last day or two, which is refreshing. But it hasn't gone away. November is a very important month in COVID times. 2019, probably when the virus was first identified in Wuhan. That's a horseshoe bat. 98% shares the virus, because that was flying in the caves of southern China, and that's probably the basis of the virus. 2020 November, the first vaccines, as quick as that. Quite remarkable. 2021 November, Omicron changed everything. Can't underestimate how important that is, and that will come up in my talk quite a bit. And then, of course, 2022. And what will we have? We don't really know. There's a few bubbling things. But for now, well, we have this at least, don't we? We have our 10th anniversary. So that's why it's an important month. And what I want to talk about is antibody immunity against all these coronaviruses. I'll explain the virus and the name. And then we'll talk about risk groups. There are people still highly vulnerable. Talk a little bit about therapy. And then talk a little bit about what we've learned from this for antibodies in general. So nothing makes sense without evolution. We've had evolution all day, haven't we? And there we go. Believe it or not, we've had B cells or adaptive immunity for around, well, 500 million years. We've got obviously a vertebral column. And those vertebrates who don't have a jaw for some reason, like lampreys, they don't have B cells and T cells, but our jaw seems to have co-evolved with B cells and T cells. You can't live without them. And so broadly speaking, very simplistically, we have these two broad arms of immunity, cells and antibodies. And I've never been to a meeting with so many Nobel Prize winners, which is great. And I'm going to bring another one in, well, another couple actually. 1908, Nobel Prize for Medicine went to these two characters, very, very important names. You'll know them, of course, Paul Ehrlich and Mechnikov. Broadly speaking, they sort of stem this divide between serology antibodies of Paul Ehrlich and Mechnikov, who was cells. He was the guy who showed phagocytosis. Not really a T-cell immunologist, but he was the cellular immunologist. He didn't even, there was a lot of immune wars at this time. He didn't even turn up for the 1908 presentation, Mechnikov. So for the moment, although actually cellular immunity is very much my expertise, probably, we're going to say goodbye to Mechnikov, at least for now, but I'll bring him in later. But we're going to focus on antibodies for the next few minutes at least. So, coronavirus, as I say, not really got that much attention in the last day or two. So where did they come from? Well, the first coronavirus was isolated from a boy in a private school down in southern England back in 1961. Quite an important year for me, actually, so there you go. But and it was a highly contagious virus because they had a research unit for the common cold, and people used to go and spend their summer holidays in this unit and get infected with these common cold viruses. And they didn't know what it was, couldn't culture it for